Welcome to the 2023 edition of Trieste Next. I'm Luca, an AI-generated speaker who will be presenting our festival. In the next 3 days we will be talking about a new world. This is the title of the 12th edition of our Festival of Science. Innovation and research, the ethical limits and new frontiers of science will be the center of all our events. What will the new world be like? Will we be able to face the challenges of the near future applying new scientific discoveries and technologies in a sustainable way? This will be the underlying theme of Trieste Next, with 100 events in English and Italian, the Science Book of the Year Literary Award, 300 speakers, 70 activities for schools and 40 exhibition spaces that are open for everybody in the city center close to the town hall. Before leaving the floor to our next panelists, I would like to thank all the scientific institutions within the Trieste City of Knowledge Network, the main partner together with our content partners and sponsors for supporting our project. Mostly, thank you to the 200 volunteers who are helping us. I have one last remark for all of you. The next event will last 75 minutes. Our speakers will answer all of your questions in the final 15 minutes. Also, please remember to check the updated schedule of events on our website. Thank you and enjoy our festival.
All right, good morning everyone, and welcome to the poster presentation session for the Biotechnologies disciplines. We have 4 speakers from various specializations within the biotechnology discipline presenting their research work with us today. And the format for the presentations will be 15 minutes for each speaker. And by the end of the session, we'll have 15 minutes for everyone to ask the questions to the speakers. So first of all, we have Aishwarryaba Jadeja over here. She's a master's student in Bioinformatics at University of Saarlandes in Germany. And she's also a research assistant at the Korean Institute of Science and Technology.
Good morning to one and all present here, and today I would like to talk about my research work which I have been doing for like past 2 years at my workplace. So that is like fabrication of biosensors in the microfluidic channels, and so basically it's more of a biotechnology or biomedical application so there are like currently some of the areas where I would like to talk about. So currently, there are many techniques available for the detection of diseases, like ELISA, which is like it takes 4 hours for the detection. There is mass spectrometry, which takes around like 4 days for the detection. But then we need something which is specific, fast, and accurate, as well as with less economical damage. And in that situation, we were looking for some aspects where we can work with them. And so we decided that we should go with some of the techniques which are like, we can use it as biosensors. And so this is my work. So what we're gonna do is that we fabricate the nanowires on the microfluidic chips, and that will be used for the detection of the diseases. And for this specific techniques, so we have like different conductive polymer molecules which we used, and for that it can be polypyrrole or P-dot. Both are like good with which provides a high and condensed nanostructure, and that we used for our studies. At the same time, we also used some photoinitiators like AGNO3 and LEP, but at the same time, we also used some of the copolymers and co-oligomers like PEO and other things. So these are the chemical molecules which we used for our studies. And so the whole methodology goes like, as we would want to specifically focus on detecting the diseases faster and accurately. Therefore, we are using some of the high-tech methods like 2-photon polymerization, where we are highly, so as we can, if we see, oh my goodness, sorry about this. Okay, so here if we, yeah, come on. Yeah. So if we see here, this is like the main region as of like where we are focusing with the photons. So this area will create a patterns like this, and these patterns are specifically written on the microfluidic chips, which will be used for the detection of the diseases. So what we're gonna do is that here we're gonna use some microfluidic channels. So we prepared first. In the starting of the experimentations, we prepared the microfluidic chips. So we fabricate this kind of chips, and they have 2 electrodes in the starting and at the end. And so with these 2 electrodes, we also have this, after the next step would be passivation layer, where we coat the whole specific microfluidic channel with the PEG layer for the so the PEG is a grafting copolymer which helps to put the main nanowire there, and then we fabricate the nanowire patterning, and in that specifically we not just only put the nanowire, but we also put the conjugated beads, so that those beads will help us to bind the specific antibodies on the surface. So if you know, here we can see that here we have this the glass substrate, but at the same time we have this conjugated beads, and there we're gonna find and bind the specific antigen correlating to the specific disease. It can be any disease. So we have specific antibody extracted from it, which we're gonna bind it here. So we have this antibody binding to the target specific, and after that we're gonna put it with the serum albumin to avoid the unwanted or non-specific bindings. So we're gonna stop it there, and then we're gonna use this to detect if so we're gonna perform set of tests. After this whole procedure, it can be like with the electron microscope, the SAM images, the conductivity image measurement. It can be with the AFM. This is the atomic force microscope, which we use to check the height of the surface and how much it is changing. So that is the thing. And this is the whole our experimental setup. And here we start with this laser. So we have like correlation with the University of Saarlandes. They have specific lab for this kind of instrumentation, where they have this instrument where we can put this whole chip on. So basically we're gonna have chip at the end. We're gonna pass the laser. It will go from this whole layers and it will come to the end. And then it will distracted into 2 things. One is with our camera, and another one is with the chip. So there it will go and start writing. So the start and the writing of the nanowire would be in this direction, or it can be like this. So now there will be a question as of like why we use 2 different conductive polymers. It can be polypyrrole or PE dot. So basically with the polypyrrole, when we were working with that, we had some complications as of like the patterns were not dense, they were quite sparse, and the pattern writing was not thin. So that was the reason why we wanted a thin pattern, so that detection would be very precise and accurate. And that is the reason why we created into the specific region, and that's how it is like at the moment with the PE dot. And with this specific region, so we have around, like we tried with the first. So this is like the main biosensors, but we didn't work with that specifically. We worked specifically with the normal chips, which we fabricated, which is not here actually. But with that, there was like around 30 channels. So for the trial and error purpose, we used those 30 channels, and we measured this specific region where we had changed a few of the parameters accordingly. So if the pattern is like dense, if it is not dense, then we need to change the laser power. It can be like what kind of laser type is there. So we changed the parameters accordingly to get the specific pattern. And after getting that, we still focused on the other side as of like for the measurement. As I said, this is the AFM results, like the atomic force. And here we get the results as of like, so the height was not how we wanted it to be. It was like around very less, and 1.1 nanometer, and it was not good result. So we are trying with different photo initiators at the moment to try and improve the patterning, and as well as we are also try and changing some of the conductive polymers, copolymers, to improve the results. This is the conductivity result, the IV IV result with the P dot, and here we can see the pattern is, so here with the polypyrrole the pattern is quite sparse and not condensed, but here it is quite condensed and it has a proper result. So that is the reason why we are using P dot at the moment. And so basically here is the results from the same image, which is the electron microscope, and here is the conductivity results, and we can see the conductivity data going high, so it indicates that the pattern is conductive, and we can, so if there is a, so if the antibody is binding there, that means there will be a vibration and change in the conductivity, and that we can detect easily with this specific technique. And so that is the one of the things. And then we, so basically after doing the list of experimentation with this specific thing, next thing was working with the biosensors. So we used 2 different set of hormones for the detection, and we can use any of these disease antibodies, but we can also, we wanted to work with the hormones, and that is the reason why we used 17 HSD and testosterone. And in that specific work, what we did was that we fabricated the specific chip like this, and that we put the samples. So here we have different layers. First will be like, so the last 2 are the reference, this will be blank, and the first 3 will be with the sensors. So there we will have our hormones, like put in, hormones antibodies put inside, and then we're gonna check with the, we're gonna minus it with the blank, and then we're gonna see what changes are happening in the conductivity results. And if there is a change, that means they were successfully able to detect the variations, and that's what we found here. So here we can see that the graph is going up, but for some of the blank, there was like no much variation, which indicates that with the blank there is no antibodies. So we can differentiate the, like the difference between if the antibody is bound to that specific region or not, and if there is a variation or not. So that's what we did here, and that is also one of the results. So basically from, like we have been working for like more than 2 years, and in that specific analysis we come to conclusion that we still lacking that condensed patterns which we want, and the patterns are not to the accurate point of how we want. So if we want to get the accurate results with regards to cancer or any other diseases, that means we need to have the nanowire pattern in such a way that it will not lose the binding. It should be condensed, it should be fabricated properly, and it should have good results. So here we can see some of the variations, and during the results also, like the experimentation also, we face a lot of complications with regards to laser writing, as well as because we have to do the experimentation in a different division of the steps. First we need to prepare the chips, then we need to go for the laser writing. So during this, any of this procedure, if there is some error with the pattern fabrication, then it will be a failure of the experimentation. So that is the reason here it's pretty difficult, but so that is the reason why we are working on this one. And so at the moment we got the results with the successful result with the hormones like 17 HSD, and we will see how it's gonna go with the further experimentation. So the research is at the moment continuation, and yeah, so that is it. Thank you.
All right, thank you, Aishwarya. Up next, we have Malgorzata Adamiec-Organiściok. She's a PhD student in Biomedical Engineering at the Silesian University of Technology in Poland.
So, good morning. I'm a PhD student at the Silesian University of Technology, and I want to present the results of our group project. The project is about regulation of programmed and unprogrammed cell death in various cell lines. So the cell death, it's a very important process in our body, because when something is damaged and the cells is not functional good, so there is the defense before the consequences. Regulated cell death plays a significant role in the body homeostasis, both in physiology and pathology. Excessive or insufficient regulated cell death may cause disease such as neurodegeneration and cancer. So we have a few regulated cell death. For example, it's necrosis, necroptosis, apoptosis, or ferroptosis. Ferroptosis is not an apoptotic programmed cell death dependent on the iron accumulation, and it's characterized by accumulation of lipid peroxidation product. Its initiation by failure of defense mechanism, including glutathione-dependent antioxidant. So in resulting in uncontrolled lipid peroxidation, the cell is death. We do the experiments on the few many cell lines in the different tissue and different region of the body. But here I will present only the skin cell lines. So the results was on the keratinocytes, normal skin cell lines, melanoma 1205 Lu, and GBX4 knockout keratinocytes obtained by CRISPR-Cas9 method. Cells were treated with Arrestin with the two doses, 5 and 10 micromoles for 24 hours. We measure a global reactive oxygen species, superoxide ion and nitric oxygen. We check the apoptosis level, and this is performed by flow cytometry. We check the cell viability and lipid peroxidation by calorimetric assay, and we measure the mRNA level of genes related to the ferroptosis and oxidative stress pathway. So Arrestin is a small molecule capable to initiating the ferroptosis cell death. She functionally inhibits the cysteine glutamine antiporter, and because of that we don't have enough cysteine in the cell, so we don't have enough to produce new glutathione. So the pool of glutathione in the cell, the most important antioxidant, it's depleted, and it causes the uncontrolled lipid peroxidation, and consequently the cell death. So we measure the reactive oxygen species in the two cell lines. First in the keratinocytes, the normal cell lines, and the cancer cell line. We observe that both in normal and cancer cell line the level of ROS is increased, and the question is which oxidant it was. So we check the superoxide ion, and we observe that only a little increase in the keratinocytes, and there is a decrease in the cancer cells. So then we check the nitric oxide. We don't see any fluctuation. There is a very similar level in both cell lines. So we make the MTT test to see the viability of the cell after adding the inducer of ferroptosis. And we observe that the normal cell, looking like it can be resistant for this activation, this cell death, because the decrease is only to viability is increased to 80%, and in the cancer cells it's like 20% viability after adding Arrestin. So the question was if we don't turn on the apoptosis or necrosis. So we check it by the flow cytometry, and we don't observe the difference between control and after treating cells in the level of necrosis or apoptosis in both cell line. So then ferroptosis is for the accumulation of lipid peroxidation product, and we check the lipid peroxidation in the cells. So in the normal cells, the result is presented by the ratio of reduced cells oxidation to signal to oxidized signal. So if the chart is bigger, we have more reduced lipids. So in keratinocytes, we can see that after treating Arrestin, there is no the level of oxidation is not increased. It's more reduced cells than oxidized. We do the confocal microscopy, and the green signal is from oxidized, red is from reduced. So here is the merge picture, and we can see that green with red merge, it's yellow, so we can see that it's no a big difference between control and the highest dose of Erastin. But in the cancer cells, we can see that after treating, we have more oxidized lipids than reduced. The chart is lower. So here on the confocal microscopy, we see that after treating the Erastin, we have more green signal in the cells. Then we want to check the marker genes of ferroptosis, and the one of important is ACSL4. It's the marker of ferroptosis. When it's increased, it causes us that the cells are sensitive. If it's decreased, it's for the resistant cells. It's important because he provides the lipid oxidation and changing the lipids for oxidized lipids. So in the both cell line we measure the expression, and in keratinocytes we see the decrease of the expression level, and cancer cells we see increase of the expression level. So after that, we check the transferrin of receptor of ferritin. This is the receptor that iron coming into the cells, and then in Fenton reaction making the lipid, the oxidants, and provided the lipid oxidation. So in the normal cells, the level of expression of transferrin receptor is decreased, and in cancer cells it's increased. From the literature, we know that some cells can be sensitive or resistant on the type of cell death, and from literature we know that sensitive cells has increased expression of these two genes, and resistant don't have expression or it's silence. So this results making us question if our normal cells can be resistant and cancer can be sensitive. So we can use this inhibitor to turn on cell death only in the one type of cells, not in the two cells. So we measured two more genes. It was PROM2 and NRF2. PROM2 is a gene that it's telling us that cells are resistant for ferroptosis. It's not expression in the cell which are sensitive. So in our normal cells we see very intensive expression of this gene, and in cancer cells we don't see any expression. About the NRF2, we see increased in both cell line, but in keratinocytes only in the lowest dose. In the highest dose, it's increased, and in the cancer cells, we see increased in the both dose. So after that, we want to see how cells look like after the treating, because from literature we know that Erastin can broke the membranes of mitochondria. The mitochondria does not look normal. So here we have the TEM microscopy and morphology of the cell. In up we see the keratinocyte. It's a normal mitochondria in the cells. And after treating Erastin the dose of 5, we can observe starting the mitophagy around the mitochondria. And after the dose of 10 micromoles of Erastin, we can see the crystal circulation and mitophagy, vacuolization of the mitochondria, and And in the down, we see the cancer cells, which even in the dose of 5 micromole, they have the damage. So normal mitochondria looks healthy, but after adding the arrestin, the dose of 5, we can see the vaporization of mitochondria and the mitophagy, the starting mitophagy. And in the dose of 10 micromoles, we can observe that the mitochondria is broken. They are circularization of the crystal, degradation of comps, and mitophagy and vacuorization. So after that, we know that HACAT can be resistant for the ferroptosis, and cancer cells are sensitive. So we want to know if we delayed the most important gene in the cells, the GPX4, because he is the most important antioxidant. He turned on the peroxidized lipids again to the not oxidized, but reduced. So we use the technique of CRISPR-Cas9 and cut off the gene of GPX4. After a few experiments, we see that on the Western blot that we don't have the protein. So then we check the viability of cell after treating of arrestin, and this cell then normal. So the level of viability is increased in the highest dose a little lower, because it's something like 70% viability, and normal cells have only the 80. We measure the gene expression of markers also, so we see no expression in the transferrin receptor. In the lowest dose, the higher expression with the highest dose of arrestin. The ACSL4 is not expression again in the 5 micromole of arrestin, and higher expression in the dose of 10 micromoles of arrestin. We don't see the expression of PROM2, so now we don't have this marker of resistance cells. And the NRF2 is increased. We measure the lipid peroxidation in the cells, and under confocal microscopy we can see that even the normal, the control cells have a lot of green signals, so a lot of oxidized lipid, and a small amount of the resistance, reduced lipids. After adding the arrestin, we see more green signal, more oxidation, and after biggest dose there is less cell after treating. So and the signal is very high of the green. We also measure the iron because of the so we see that after gen modification, we have iron accumulate in the cell, and in normal cell we don't see that. The the purple signal of iron. So tested skin cell line present opposite sensitive to ferroptosis. Keratinocytes cells are less susceptible to ferroptosis than cancer cells, which are more sensitive. Arrestin induced cell death in keratinocytes and LU are neither related to the apoptosis or necrosis. GPX4 knockout cells no expression the transferrin receptor, and CRISPR modification results is changing in marker gene expression level, but no impacted cell viability. Thank you.
Thank you so much, Malgorzata. Up next, we have Meghan Manor. She's a master's student in biotherapeutics at University College Dublin in Ireland.
Hi everyone, just waiting for the, good, okay. Where do I point? Okay, there we go. So this is my poster. I'm just gonna go walk through the project that I did in my master's. We worked on expressing and purifying an Alzheimer's associated protein, beta-2 microglobulin, and worked on identifying binders derived from a library called an SX-K-mer. So beta-2 microglobulin is a small protein. It's only about 12 kilodaltons, and it's part of the extracellular component of a major histocompatibility complex. So it's a major component in immune response, and therefore misregulation is often resulting in immune diseases such as cancer. So only recently it has also been associated with co-aggregating with the Alzheimer's associated protein amyloid beta 42. And this co-aggregation causes thicker amyloid fibrils than you would see with just amyloid beta on its own. So this is why we chose this protein. It's very common in the body and potentially an easy target for therapeutic approaches. So our first step of the project was to create a construct that we could work with in a soluble form. So because beta-2 microglobulin does aggregate into amyloid fibrils, we had to add some components to it to make it more soluble. So the M-cherry, which is a fluorescent tag, much like GFP, we used that to help solubilize it and also to help visualize it during the project. And then we had a histidine tag at the end for purification. So then we expressed it in a T7 expression vector, which basically allows us to control the expression in E. coli using IPTG as an inducer. So without the IPTG, the cells will just grow as normal without producing our protein. And then when we induce it, we can get higher amounts of our protein being produced. So we worked with this. We had to optimize for temperature conditions, IPTG concentration, the length of expression time, and the batch size. And then once we had done this, we harvested our cell cultures with sonication and took our soluble extracts and analyzed that to see where we were getting our protein. And then we did 2 purification steps. We did immobilized metal ion chromatography using the histidine tag, and then a size exclusion as our final purification. So this was our main result of the optimization component of our expression. We found that long expression time, 18 hours, and 800 milliliter cell culture produced very good results before we started with 200 milliliters and shorter expression. And we were getting some results, but we wanted to see if we could scale it up, and it worked. So this particular SDS page gel was after our IMAC step with a 18-hour expression and 800 milliliter culture. And we found a lot of expression of our protein here. So the different temperatures, these are all 30 degrees with different IPTG concentrations. This was 25 and this was 20. So basically we continued working with 30 degrees because it's easier to work with, and we were getting similar results with all concentrations of IPTG at that temperature. So then once we had our purified construct, we immobilized it onto immunotubes to perform a phage display campaign. So basically we added a phage library, which allows us to present like a massive amount of variability in the sequences of the protein, so we can screen for the best binders. And so we immobilized that onto immunotubes, and we allowed the library to bind and washed out any non-specific binders. Then we eluted our phage and infected it back into a TG1 E. coli bacteria, and with the help of helper phage, we amplified our signal, and we did this 3 times consecutively, using the library from the previous round to help enhance the specificity of our library. So we used a combination of 2 libraries called SX-K-mer-Side and SX-K-mer-Loop, which I'll go into a little bit more detail in a second here. But then while we were doing the phage display campaign, we monitored the amount of phage we were recovering. So we counted the titers of our phage, and we found moderate amounts with each round. We didn't get an increase, which is what we would have expected, but because we were still getting phage in each round, we continued on with the project. So we had to quantify the strength of our binding of these binders using ELISA. So we used an indirect ELISA, which I have a little schematic here. This is our protein immobilized onto the wells, and then we would add the phage, wash out any non-specific binding, and then the antibody reporter, which allows us to visualize the reaction. So we did 2 different ELISAs here. So the first one was a polyclonal format. So we took a library, a small amount of the library from each round, and measured the binding against our construct as well as BSA for control. And we also took some of that library and bound it to the wells as well to see if we could get anything, because basically what we found is we weren't getting any color change on the construct or the BSA, which was a little confusing. But we were still getting color change with our phage-coated wells. So we continued on to a monoclonal ELISA in order to try and amplify unique binders instead of all of them at once. So we took individual colonies from our last round, and we let them grow, amplified their phage, and then eluted that and performed the ELISA again. So with this result, we got a lot of color change this time, and this graph is our top 20 based on purely on absorbance levels. This blue line here is our threshold that we calculated to be 3 times higher than the average of the blanks. And so everything to the left of that red line was taken for further analysis, which we started with sequencing. So moving on to our next step, I'll go into a little more detail on what the libraries are. So phage display is mostly done with monoclonal antibodies. So they take the variable regions of antibodies and screen that. We used a different type of library. So it's made of a Calbindin D9K scaffold, which is basically just 2 EF domains together. So the first one, the side library, has 6 variable amino acids that are shown in the rainbow colors there, and they make a nice little surface along the side of the protein. The substitutions in the sequence are right there. So that's the first one, and it's in the EF1 domain. And then the loop library is actually an insertion of 7 random residues shown here, and then they're flanked by 3 glycine residues on either side to help make that loop that kind of sticks out from the protein. So we used sequencing and then converted that to amino acid sequences to see if these 14 clones were producing the library and what their variable sequences were. And then we modeled those proteins using Swiss model into 3D structures. So the results from that, we actually got 12 unique sequences that were characteristic of our libraries. However, one of them had the result from the sequencing had a mutation in the variable region, so we couldn't really confirm what the amino acid sequence was from that. So we eliminated that one. So we had 11 unique clones that we could actually work with. 7 of them came from our side library, and these are shown right here with the different variable amino acids highlighted, and the different colors are just the different characteristics of the amino acids. And then we had 4 of our loop library, which are shown here. And so then with the 3D models, the one on the left here is an example of our side library. And so it was just modeled based off a template of Calbindin D9K, which was the scaffold of the protein. And then the variable region is highlighted here. So I used clone F5 to do this because it was a little interesting to me that that one had a lot more hydrophobic residues. So in the first two positions. So just something interesting about that one. And then the loop library, we used clone G2, which had an alternating pattern of hydrophilic and hydrophobic on the sequence in the first 5 residues. So that shown here, the blue is just the glycine residues, so those variable regions are highlighted there. And on both of those models, we could clearly see the binding surface of the proteins. So in conclusion, we were able to express the construct in a soluble form and optimize that for phage display. And then with 3 rounds of panning and monoclonal ELISA, we successfully found 11 different unique sequences that had strong binding to our construct. But we do need further characterization of the binding against like irrelevant proteins such as BSA to eliminate non-specific binders and variations of the construct such as something with mCherry and the histidine tag without beta-2M, just to determine the specificity of our binders towards beta-2 microglobulin specifically and their potential as therapeutics. Thank you.
Thank you, Megan. And last, we have Soraya Fraga Pampyn. She's a master's student in biomedical research with specialization in health biotechnology at the Department of Microbiology and Parasitology at University of Santiago de Compostela in Spain.
Well, thank you, everyone. I'm going to switch completely the theme. We are going to talk about bacteria that actually affect fishes, but they can be translational to, well, they flow through, can be used for also bacteria that affect well, humans. So my work was about unraveling the sequence that govern the replication of an unstable plasmid that is in the genetic structure of a marine bacterium that is pathogenic for fish. So, sorry. So the content, don't get scared. I'm going to go through really quick. I just don't like a lot of letter into the same presentation, so I'm going to be really quick. We are going to do an introduction to be like everybody in the same page to understand the rest of the presentation. The objectives, that are 3: studying the curation rate of the PPH DP70, that is my plasmid. Then studying the incompatibility with another plasmid that is MDR, that means that is a multi-resistant plasmid, and then the identification of the origin of replication, that is the main thing that is responsible for this instability, and then a few conclusions. So this is the bacteria that has the plasmid, is Photobacterium dancellae pisticida. We are going to refer from now on from this bacteria to PDP. It affects those fishes that in aquaculture they are highly valuable. So it causes photobacteriosis, it affects an enormous host range of fishes, it has a very big mortality rate, and the distribution is ubiquitous. So it's like actually a real danger. Talking about genetics, it has 2 chromosomes and then 3 plasmids. We are going to talk about this one that is not the one that I'm studying, but just to use it as a genetic marker, so we need to know what are we talking about. And then this is my main focus, this plasmid. So regarding to the first plasmid, we are going to use this genetical marker. It has a virulence factor and is prevalent, and it has a lot of stability, so we can use it just because of these qualities to be a good genetic marker to know if we are working with the bacteria that I just talked about or not. And then about the pathogenicity of this toxin. It eliminates phagocytes by secondary post-apoptotic necrosis, but it's not going to affect my work at all. This is the plasmid. I want you to pay carefully attention to these symbols. Those are islands. That means they have like a repetition of sequences, and this is going to make my work like very difficult, because when you need to like design all these how do you say that, primers for doing PCR, is going to be really difficult to optimize the technique. So we are going to use a marker to track this plasmid that is going to be called FRPA, and this is a virulence factor that is piscibactin. This is not going to affect my work either. Plasmid curing is when the plasmids are eliminated by the bacteria, but the bacteria are still alive. So that was the process that we need to track in my work, and it has implications in your daily work at lab, because you're trying to work with a bacteria that is a pathogen, and if it doesn't have this plasmid, it's not going to be pathogenic at all. And then if you're trying to do vaccines to vaccinate those fishes, the virulence factor is on this plasmid, so you're not making a vaccine that is actually working for that. Basically, you're wasting money. So 3 main objectives: quantitative analysis of this loss of virulence of this plasmid, because we were doing successive passages and we were basically showing that this is happening, like this is curing with the passages. Then the study of the incompatibility with the MDR plasmid that has an homologous replication system, and then discovering what are the genetic sequences that constitute the origin of replication. With the first one, the curation rate of this plasmid. So we start with a colony. It has the 2 plasmids. Then we just take a single colony. The idea was to have less like variability, but I'm going to show you later that single colonies are not homogeneous as we thought they were. Then we incubate that overnight, and then we do serial dilutions and PCR with single multiplex PCR, and then we are going to do this passage after passage to see the curation rate. So we start with the first passage when we just defreeze the bacteria and we just do this first passage. So from our master collection, we start with a curation rate of 14%, that is massive, and then with all the passages it's going to go down to 52. So with only 4 passages, we are losing more than half of the plasmid. Then we are going to go to study the incompatibility with another MDR plasmid. So there can be a lot of reasons why 2 plasmids are incompatible. One of these ones is because they have the same origin of replication. So if you have multiple proteins that can recognize different origins of replication, everything is going to be fine because they are going to be replicated by themselves and they are going to be conserved in similar rates. But if you have only one machinery to do all this replication, they are not going to distinguish because the origins are the same. So you're going to lose one of them with the time. And then there's partitioning mechanisms, because some of the plasmids can go just the cell cytoplasms can just disperse the plasmids that are on one side on the plasmids that are on the other side, but these plasmids has partitioning mechanisms that are like normal mitosis but for bacteria. So we compare both sequences, the 2 plasmids, and they are similar in these 3 regions that we are going to use later as a putative origin of replication. And the thing that we do, it was a really easy technique, is basically bacterial conjugation with markers for those plasmids that are antibiotic markers. So then after 48 hours, we are going to select this strain that has this plasmid and loses the plasmid that is my plasmid of study. That shows that they are really incompatible and they cannot be in the same cell at the same time. So as we see, TEDB will track this plasmid, the one that we were expecting to have on our bacteria, and then we have the FRP that was supposed to be on my plasmid that we want to lose. And as we were hoping, we have all of these plasmids on our bacteria, and we lose the plasmid that we were working with. That means that they cannot be on the same bacteria, so they are incompatible. And then after that, and using this genetical comparison, we are going to work with these 3 sequences that they are named here, region 3, REPA, and NONACA, as putative regions that are involved on the replication. That was the main objective of my work. So for the replication of plasmids, there are a lot of literature, and they say that REPA is normally a main component of all this replication system, and then they are going to try to find iteron sequences, and we have 2 putative iteron sequences, so we are going to shuffle them to find the minimum sequence that is in charge of the replication, basically. That's a lot, but the main thing about that is all these experiments, all the dots are going to be trans regions. That means that they don't need to be in the same molecule to play a role. So they don't have this physical restriction or being together. And those squares are the regions that we are going to name as cis acting, because they need to be in the same molecule to act as they are expected to do it. Basically, if we are putting sequences on cis acting, we are thinking that this sequence is the origin of replication, and the other things are necessary to do this replication, but they are not the sequence that is in charge of that. And then all these results until we get to this one that is going to be the minimum origin of replication. Basically, it's the same. We started with the most simple one. We had like previous results, like just they didn't work, but we need to test it again because there can be ambiental conditions or wherever, so it didn't work. So after trying this one, we are going to start doing sequences in trans because maybe this sequence could be the origin of replication, but needs another thing that is on the other part of the plasmid to work. So we actually start putting on E. coli all those trans constructs, and then doing heat shock to put all these things constructs. And it didn't work when we put REP-A, and we were expecting that to work because REP-A, as I told you, there's plenty of literature that they're saying that is involved in replication. So there was 2 iterons regions. This one didn't work, so this is more small. It's going to be easier to do all the cloning, so basically we keep with this iteron region because the experiment could be faster. Same combination, we just changed this. It didn't work. So we are starting to add everything that is possible. So it has to work, right? Because there's nothing else to put there. It works, but we now are aiming to reduce to the minimum expression this origin of replication, because this is going to have like multiple applications later on. We delete REP-A. We are not hoping that this is not going to work because REP-A is on the literature, like you need that for the replication, so it didn't work, but we need to test that hypothesis. Apart from that, we also want to put the cis acting region shorter. So this is the cis acting region. It has a lot of proteins, mainly this one. So we didn't expect that, but actually the intergenic region was the one implicated on the replication, so we are going to test this region. We did the same with only this region. It didn't work because it didn't have REP-A. We expect that, but we need to test this hypothesis. We went back to everything together, but now with this region shorted, it works, so now we have this region completely shorted. We need to shuffle the other regions to find the trans-acting region, the shortest trans-acting region. We delete region 3, it works because it has REPA, so that was the minimum origin of replication. It has non-ACA only within the intergenic region, and then REPA that is involved on the replication. When we got to that point, some people can think that PCR is a really good technique, so even with a 1% of positive clones, they are going to have like a positive signal. So we did a genetic confirmation, basically plating bacteria. When we did that with this antibiotic was selecting the trans region. So you're starting plating the trans region. This region, if it's not necessary for replication because it was in trans, is not going to be when you put the 2 antibiotics. But it shows that 100% of the things that you're only selecting the cis region, they keep trans region. So that shows that basically you need this trans region. When you do the opposite, when you keep this trans region, it's not necessary to keep, like REPA can live without the origin of replication, but the origin of replication always needs REPA. That's the main thing of this contribution. This is not the end yet. I still have the conclusions, but basically, as I told you before, optimizing all these primers was massive work because there's like a virulence factor that is almost the whole plasmid. Then you have intergenic regions that are maybe another half, and then a lot of islands of pathogenicity that they are repeated sequences. So if you have a GC percentage that is awful, and then the rest of the plasmid is like repeated sequences, it's really difficult to get a target plasmid that actually is specific to this region. And the cloning, every time that you're seeing transacting regions, you need to do competent cells. Like there's a massive work more than shuffling sequences behind. So conclusions, 3 main conclusions. The virulence plasmid suffers in vitro instability, as I showed you, starting from 14 in the first passes to more than half of the, like we lose more than half. So this could be a thing that you need to take in account in laboratories that are working with vaccines, because you're not working with a pathogenic bacteria and you're selling that. To aquaculture factories, the minimal elements required for the replication are those 2 distant regions that are non-ACA minimum, this intergenic region, and then REPA. And then this plasmid was incompatible with the MDR plasmid. That result may contribute to further design of strategies to control the spread of these plasmids, and also do some biotechnological vectors like suicide vectors, or maybe being able to literally transform those strains instead of having like a step in the middle going with E. coli. That's a main issue, really important also in the field. And that's it.
Thank you so much, Soraya. So now we have about 15 minutes in case there's any questions from the audience.
Okay. Should I hold it or? Okay. Thanks, guys, for the talks. It was super incredible and very extensive in terms of fields of science. One question for the biosensors, because I think this is the field where I know the least amount of things. How do you go about having an antibody and in silico chip? How do you go from I detect an antigen and transform this into an electric signal? How do you go about this? This is what I would like to know. Thank you.
So basically we have this chip which has the nanowire where we are putting the bits. On top of that we are putting it for the binding of the antigen or the antibody, and then when it binds there, so we are checking the measurement of the conductivity. So if before the binding there should be a stable conductivity, and when it binds there should be a variation, because all the particles, they have some sort of charge, and when it binds there should be variation in the conductivity. At the same time we are doing a lot of other things like SAM images where we are checking if there is some binding, then there should be variation in the pattern. So that's also one more thing. And we also doing the AFM, the atomic force microscope studies. So in that we are measuring the height of the pattern, so it should be stable, but when there is variation, we can detect that there is some changes. So our primary focus is like based on the conductivity results, but then we go further for the binding check, and it takes only fraction of like, so when it binds there and when we measure into the conductivity just take like around few minutes. Whereas the other test like ELISA and mass spectrometry, it takes days and hours. So if we want a results, if someone is suffering from cancer or any kind of heart disease, if we want to know like if the person is having heart disease or something, we couldn't wait for hours, then that person might have died. So it's like a fast, that is the reason why we are doing this one. Yeah, thank you.
Any other questions apart from this? Thank you so much, all four of you. It was really good session. My question is from the candidate from Poland. I'm sorry, I don't know how to pronounce your name. So yeah, you were talking about apoptosis. So firstly, I just want to ask, what is the take-home message about your project? Because that was not really clear for me.
The common message is that we can control the cell death, so we can turn on the cell death probably in some cancer cell line. Don't disturb the normal cell line. So we can use the regulation to future clinical.
And one more question. Your data was really interesting. I just want to ask, the graphs which you had made, which test you have used? I mean, the graph, you have asterisks on that. What was that? Which graph? Every graph. I mean, you have you have beautiful graphs, and you have asterisk marks on that. So what was the test? What was the, I mean, did you done the t-test or what was it?
Yeah, it's the t-test.
Okay.
T-test.
Okay, that's it. Thank you. Thank you, everyone. All right, any more questions? I don't see any, so I suppose this concludes the event. Thank you so much, everyone, for sitting through the talk. And thank you so much to all the speakers for presenting their research work here today.